నమస్కారం. డాక్టర్ బిఆర్ అంబేద్కర్ సార్వత్రిక విశ్వవిద్యాలయం విద్యార్థులకు స్వాగతం. ఈ ప్రసారంలో
Welcome to the students of Dr. B.R. Ambedkar Open University pursuing B.Sc Drug Science, and today's lesson is Morphology and Fine Structure of Bacteria. This is a lesson from Microbial Biotechnology. This lesson will help the students to understand the important characteristics of bacteria. As you know, bacteria are small microorganisms with microscopic structure. The bacteria are millions of bacteria are present in the atmosphere. They are in the soil, they are in the water, they are in the food. So all these bacteria, they play a paramount role. All the bacteria are not harmful. Only pathogenic bacteria are harmful. Pathogenic bacteria are those which produce disease, but all otherwise bacteria play an important role in the environment. For example, you have bacteria who are called decomposers. The food chain, you have producers, consumers and decomposers. So the decomposers, either animals or human beings, the dead particles which are buried in the soil are converted, they are purified by the bacteria, and they are converted to important inorganic constituents like nitrates. So there is a nitrogen bacteria. So bacteria plays an important role for environment, but bacteria occur very widely in nature. The smallest bacteria, the size of bacteria, morphology includes the size, shape and the internal arrangement, the cell structure. The cell structure has got an outer structure and inner structure. So bacteria, the study of all these things will help you to understand how bacteria grow and how bacteria function and various other intra-physical and chemical properties of bacteria. In this chart you will find out the various classification of microorganisms. As you can see, bacteria are prokaryotes. So you have the bacillus and you have the blue-green algae. As you know, prokaryotes are simple structure, whereas eukaryotes have got complex structure. The other protozoa, fungi, as you can see, different varieties of protozoa and different varieties of fungi. So this is the nominal classification of various bacteria that are studied, and as you know, because of the advent of microscopic techniques and electron microscope, we are able to find out the finer structures of bacteria. And then if you look at the shape and arrangement of bacteria, there are, each bacteria has got a diverse structure. Suppose cocci, bacillus, spiral, these are the three important varieties of bacteria that is commonly encountered for various environmental conditions. Cocci are spherical or oval shaped. As you can see, this is single coccus, this is Diplococci. Diplococci means two single cells are attached, that is why it is called Diplococci. Whereas this is a monococci, and then streptococci is something like a chain formation, and then this is what is called staphylococci, is something like a bunch, like a grapes bunch. You can see a formation of grape bunch, and other things is what is called tetrad. Tetrad means a combination of four individual cells, cocci, that is what is called tetrad. And then if you come at the bacilli, and you can see the bacilli are also different types of bacilli. Bacilli are rod shaped, The difference is they are spherical shaped and these are rod shaped. This is cocci, this is single cocci, this is diplobacilli, that means 2 attached, and then the streptobacillus are in the clusters, and this is like a chain, the peptides, and then this is vibrio shaped. The example Vibrio cholera. This is the bacteria which produces Vibrio cholera, and this is the shape of the bacteria Vibrio cholera. And the third one is what is called spirillum. The spirillum, as you can see, this is like a chain reaction cocci. These are spherical and oval cells arranged in pairs like diplococci or streptococci, and bacilli are rod shaped coccobacilli, and then they are examples Vibrio cholera, and then the spirals are like chain formation. And then you have different staining procedures. Depending on the stain procedure, you have gram positive and gram negative bacteria. And the second thing is the bacteria are classified into prokaryotes and eukaryotes. The difference between prokaryotes and eukaryotes are, prokaryotes are simple structures. They have got a cell wall and they are unicellular organisms, whereas eukaryotes, they are the plant cells and animal cells. Formerly there are only 2 kingdoms, the plant kingdom and animal kingdom. So, but later on there are so many microorganisms which are microscopic in size, and then later on they have identified the third kingdom called Protista. The Protista is the microbial kingdom that was announced by a scientist called Haeckel in 1886. Now all the microorganisms or viruses or bacteria, they come under this third kingdom, microorganisms kingdom, the kingdom of microbes. So now we will see the differences between eukaryotic cell and prokaryotic cell. The size, the shape, the cell structure, all of them differ from prokaryotic and eukaryotic cells. Now I will show you, this chart will tell you about the differences between a prokaryote cell and eukaryote cell. You can see the difference. Prokaryotes, and the important thing, this is a small cell wall and simple structure, the cell wall and cell membrane, whereas in the prokaryotes there is a bigger cell wall and then there is a plasma membrane, there is cytoplasmic membrane inside, and there are so many other organelles, whereas the prokaryotes will have no internal organelles. So you can see the mitochondria. Mitochondria is the powerhouse of the cell, where ATP, adenosine triphosphate, the energy is produced here. Then this is the outer cell membrane and this is the internal cell membrane. And then you have this central part where you find the nucleus and nucleolus. This is again covered by another membrane called nuclear membrane. So the nuclear membrane protects the nucleus and the nucleolus, the genetic material of the cell. And similarly you have what is called, this is endoplasmic reticulum. So this produces this. Similarly you have Golgi apparatus. Golgi apparatus is the storehouse of all the enzymes. Similarly you have the vesicles. So these are all different parts. And then the second thing is chlorophyll. As all of you know, the photosynthesis is the main criteria for the photosynthesis by plants. So chlorophyll, the dissolved chlorophyll is in the cytoplasm, whereas the chlorophyll is the chloroplast. As you know, the chloroplast and play an important role in photosynthesis. The trees, the green leaves of the tree, they produce carbon, they take carbon dioxide from the atmosphere, and then with the help of the chlorophyll and the chloroplast which is there in the chloroplast, and then the water taken from the soil, from the roots of the tree, they produce carbohydrate glucose, the photosynthesis process. So they have a bigger role of chlorophyll in case of eukaryotes. And then chromosomes, you can see only a single chromosome is present in eukaryotes, whereas this is number of multiple chromosomes are present in eukaryotes. And then what is called, there is no evidence of mitosis in this case. In the case of prokaryotes, no evidence of mitosis, only it is a unicellular, the DNA multiplication, whereas here the meiotic process of reproduction is very much there in case of prokaryotes. And then if you look at the characteristics of the differences between prokaryotes and eukaryotes, you can see the genetic material. As I told you, there is no special nucleus part in case of prokaryotes, whereas you have a membrane, a nuclear membrane is there. Inside the nuclear membrane you have what is called the nucleus and nucleolus, and they help for the replication and the replication of the DNA and then protein production. And then the genetic recombination, this is only unidirectional under a single DNA which is in the central part of the prokaryote cell, whereas here as I told you, the mitosis and meiosis are the apparatus which help for the reproduction and multiplication of the microbes. Similarly, mitochondria is present, chloroplast is present in prokaryotes, eukaryotes, whereas they are not present in prokaryotes. Similarly, there is no plasma membrane is present there, and similarly Golgi apparatus, as I told you, is the storehouse of enzymes that is present in eukaryotes, whereas it is not present in prokaryotes. Similarly, there are other inner organelles, all of them are and ribosomes. Ribosomes, this is 70S ribosomes in prokaryotes, whereas 80S ribosomes. And ribosomes, as you know, they help for the production of protein. As you know, there are 3 RNAs, mRNA, tRNA and rRNA. The ribosome RNA is the one which produces the protein. So that is not present. The ribosomes are not very much present. The differences between the prokaryotes and as you can see, the characteristic differences, organelles of genetic material is more predominant in case of prokaryotes, eukaryotes, whereas prokaryotes has a simple mechanism. And then if you look at the structure, the bacterial structure, the bacilli occur singly or in pairs, Diplococci, and then you have the bacterial structure. Bacterial structures are there which are curved with a twist, and then the internal structure, examination of a bacterial cell will tell you about the difference between a prokaryote and eukaryote. You can see the structure of a cell wall. The cell wall is common both for prokaryotes and eukaryotes, and then in this you can find the chromosomes. You have the ribosomes attached to plasma membrane, and ribosomes are there, the site where proteins are produced. You have here a capsule. Capsule also will provide a protection to the inner particles which are essential for protecting the bacteria from the bacteriophages, and this is the plasma membrane where you have, and inside you have the cytoplasm, and then you have what is called mRNA. This is the messenger RNA. From the central part of the nucleus, the genetic code is passed on by the mRNA, and then the mRNA is a messenger RNA, but it cannot translate the genetic material. You have other called tRNA, the transfer RNA, and rRNA, the ribosomal RNA, thus producing the protein. And then you have in a plant, you have a thylakoid structure here which is essential for photosynthetic apparatus. And then if you have still further felicitation or further elucidation of a membrane which is common to all the cells, you have, as you tell, this is the plasma membrane, you have another cell wall. The cell wall, it is a semi-permeable membrane. That means it allows only the essential nutrients like glucose etc. by active transport, whereas lipids are not allowed inside the cell wall. Slowly they are sent by a carrier protein, and then here you have different parts of the cytoplasm inner membrane, and you have this is second part, this is the DNA. In case of prokaryotes there is no nucleus but the DNA, so this will be useful for further multiplication of the cell. So this is how the internal structure of a cell. So the difference between the prokaryote cell and the eukaryote cell, as you can see, you have bigger multiple structures in a eukaryote cell, whereas this is simple structure, only DNA and no nucleolus and nucleus in the other substance. So that is the main difference between the prokaryote structure of the cell and the cell wall. And then you have the bacilli the bacterial structure. Examination of bacterial structure is important. You have two important points in bacterial structure. Examination of bacterial structure, the external structure and the internal structure. The external structure, as I told you, is a protective layer to see that unwanted substances like toxic chemicals or other do not enter the cell, otherwise there may be, the cell may be decayed, the cell may die. The structures external to cell wall, they are include the flagella, the pile, etc., and then the internal parts of a cell are more important, and they are structures attached to the inner part of the cell, and that makes a difference between the gram positive and gram negative bacteria. So, gram positive bacteria and gram negative bacteria, the cell wall is different. There is the combination. For example, the size of microscope, the size of microns, the microbes are very tiny. As you can see, they are calculated in what is called micrometers. That means micrometers are very, very small, 1/1000th of a meter, and then you have different bacterial structures which are essential, and the size differs, and then because of the staining procedures, some are called gram positive and some are called gram negative, the staining procedures. So, that means the staining procedures, they are simple stain, differential stain, etc., and by these stains procedures you can differentiate between gram positive bacteria and gram negative bacteria. As you can see, suppose for example, the size Clostridium botulinum, which causes food poisoning, the length is 3 to 8 micrometers, whereas the tuberculosis bacilli, the size is different. So, that is, the bacterial cell wall is important for cell to grow and develop and multiply, and the cell wall is a protective layer, and inside, in case of eukaryote cells, you have another plasma membrane which protects the inner particles like nucleolus, and the nucleus has got another membrane called nuclear membrane, so that the DNA, the nucleus and nucleolus are protected from any external injury. And then the walls of gram positive bacteria have greater amount of what is called polydiphorin, whereas in case of gram negative it is simpler, and where the walls of gram negative bacteria are more complex than gram positive bacteria. And then you have the walls of gram negative bacteria are rich in lipids, whereas in case of gram positive bacteria the bacilli are not rich in, and you have what are called semi permeable membrane and impermeable membrane. Bacterial structure is very important. Now we have seen the size, and then the structure and the arrangement, they are very important for the morphology of a bacterial cell. Examination of bacterial structure have two parts, what is called the external structure and the internal structure. Examination of bacterial cell is very essential for finding out the various components that are inside the bacterial cell and how they are protected and what are their functions. And the structures of external cell wall are very important because they protect all the material which is there inside the cell, inside the bacteria, and structures inside the cell wall, they are more important, and they should be protected from any external toxic chemicals or other bacteria. So, there are several components of the internal structures of the cell wall, and cell wall is common to all microbes, as I told you, the external cell membrane, but whereas in eukaryotes there is an inner cell membrane, there is another membrane, and similarly the mitochondria has a membrane, the Golgi apparatus has a membrane, each of them has got their own cell membrane which protect their inner components. And the cell wall, the difference between eukaryote cell and similarly the gram positive bacteria and gram negative bacteria, there are some mild differences between the sizes of different size, and both the structure and the composition of the cell wall is different from the gram-positive bacteria, and they are different from gram-negative bacteria. As I told you, gram-positive bacteria, they have a more thick layer and they have a lipid layer. As you can see, the other finer structures like plants and animals, they have a bipolar lipid layer, a cholesterol layer, and where that's a semi-permeable membrane, it will not allow all components to get into the cell, only it allows the essential parts like glucose, like amino acids, etc. They are allowed inside the cell by passive diffusion, because glucose is essential for the survival of the cell, and other parts like lipids and other things, they are slowly allowed inside. Similarly, suppose you have sodium and potassium. Sodium and potassium are essentially electrolytes, but there is intracellular and extracellular parts, there is difference, because inside you should not have more sodium and should be more potassium, and so you have a constant flow from the intracellular fluid and the extracellular fluid, and so there is a what is called sodium potassium pump, which coordinates the ratio of sodium potassium essential for the normal cell function. As you know, more sodium inside is not helpful or healthy to the patients, to the men. As you know, more sodium will increase your blood pressure, so that is why the patients with hypertension are advised to take less sodium. So sodium should not be more, and this is harmful. So that is why a ratio between sodium and potassium is to maintain. Similarly, glucose is essential for the very survival of the cell, especially the central nervous system. The brain cells require constant supply of glucose. So there the cell freely allows glucose and amino acids to get into the cell, whereas it allows slowly the lipids and other things, and they require what is called active transport. That means a carrier protein will help them to pass through the cell wall and reach inside. So the inner structures of the cell wall, there are some differences and between prokaryotes and eukaryotes, as you can see the size, and similarly the walls of gram positive and gram negative cells also there is some differences. As I told you, the gram positive bacteria have a greater amount of the peptide bonds, and then the cell wall is a composition of as compared to the gram negative bacteria, and walls of gram negative bacteria are more complex than the gram positive. Walls of gram negative bacteria are rich in lipids, whereas the walls of gram positive bacteria, they are very poor in lipids. Similarly, you have other internal structures of the cell wall like cytoplasmic membrane. The cytoplasmic membrane is not there in the prokaryotes, whereas cytoplasmic membrane plays an important role in case of eukaryote cells. Similarly, inside the cytoplasm also, so the essential components of the cell structure is very essential for the normal functioning of the cell, and the morphology, the size, structure and arrangement is very important for understanding the function of bacterial cell.
పాఠ్యాంశాలపై మీ సూచనలు, సలహాలు, సందేహాలు మాకు తెలియచేయండి. మీ సూచనలు, సలహాలు, సందేహాలు మాకు తెలియజేయవలసిన చిరునామా: డైరెక్టర్, ఆడియో విజువల్ ప్రొడక్షన్ అండ్ రీసెర్చ్ సెంటర్, డాక్టర్ బిఆర్ అంబేద్కర్ ఓపెన్ యూనివర్సిటీ, ప్రొఫెసర్ జి రామరెడ్డి మార్గ్, రోడ్ నంబర్ 46, జూబ్లీ హిల్స్, హైదరాబాద్ 500033.